Collins,
Yes, ma'am.
did you get me?
Yes, I did. I got you,
Okay.
Janaya. Thank you for checking, though. 'Cause sometimes I'm all over the place. All right. Let's take a look. So, as we talked about on Friday, we had to change our schedule a little bit because so many of you guys were behind. Right. So, let's take a look. There's not much on our to-do list this week, right? It is week 13, so we are approaching the tail end of the semester. And we've got spring break starting on Friday. And I don't know about you guys, but I am excited, but it hasn't started yet. And because we were behind last week, our exam for Unit 7 was supposed to be on Friday, and it got bumped to today. So what I'm going to do here in a second, I think it's going to make a lot of you very happy, okay? Because you'll be very glad that you attend the Zoom. We're going to go over the exam. And by go over the exam, I mean I'm going to open the exam and I'm going to quickly go over each of the questions. Now, the questions on your copy of the exam will be scrambled, right? So you got two options here. Either you just kind of like watch and listen and maybe like take some notes on paper as I'm going over the questions, or when I go over the question as I'm talking, quickly scroll and find it. I'm gonna go over with you guys. We're gonna do the test today together. All right. So for those of you at home on Zoom, you're thinking, yes. Okay. Now, when the exam is done, right, the Zoom will be over, and you need to complete the vocab for our new unit, Unit 8 on biodiversity. This is my favorite unit. This is why I became a science teacher. This is why I've studied biology my whole life. And that's what we're going to start today with the vocab. That vocab will be to tomorrow. And then tomorrow, we're going to be watching a video about sickle cell anemia. It's this crazy blood disease and its association with malaria, a disease that you get from a mosquito bite. On Wednesday, when you guys are on your own, there are two short videos. I'm talking like maybe 18 minutes tops that I'm going to share with you guys on Wednesday. There's some questions that go along with it. And I'm also going to share the EOC study guide with you. And you don't really need to work on it, on Wednesday. Just make sure that you have it. Because honestly, after spring break, I'm probably going to give you guys paper copies to work on. So I might actually scratch that until I see you guys in class on Thursday. And then obviously, 8A students, you'll have to get it when we get back from spring break. And then we have a short article about classification, and that's it, right? So really, I would say the most work we need to do is today, right? Now, I'm going to chat a little bit about biodiversity tomorrow as well as Thursday, and that really just works out perfectly because it is spring. There's going to be the insects and the butterflies and just everything springing to life, and what better way to talk about biodiversity than to actually be outside studying it. So first thing on our list is we're going to jump right to the exam. Now, I assigned your exam, so you need to go to Unit 7. So everybody needs to pull up their Google Classroom, right? You'll see it right here, third period, Unit 7 exam. Go ahead and pull that up. Now, everyone's exam might look a little different. So for those of you guys that are in here, give me a thumbs up when you've got your exam pulled up because I'm literally going to go over it with you. Okay, you good? Tay, you good? Carlos, you good? You pull your exam up? Erica, you good? Okay. Everyone at home, are we good? Are we ready to rock and roll? Now, remember, the questions are going to be kind of scrambled, so you might have to do some hunting. Either that or you just listen to what I'm saying and then just take some notes, okay? So let's take a look at question 1. What pattern of inheritance is generally associated with traits or diseases being more probable in males? So this means that men are more likely to suffer from these diseases or to have these traits, like colorblindness, male pattern baldness, hemophilia. And the reason males are more likely to have these diseases and these traits is because they are carried on the X chromosome. So this would be a female, and this would be a male. If a female inherits a defective X chromosome, the woman has a second one. However, if a guy gets an X chromosome, how many X chromosomes do we have? We have one. So if we inherit one and it's messed up, we're going to be messed up. So we call these sex-linked traits. If males tend to have the disease more than females, we call it sex-linked. Let's look at this next question. What is the most likely genotype? And anytime I say about genotype, right, I'm asking what are the genes? What is the most likely genotype you found in offspring produced by this cross? Well, there's a 25% chance of big A, big A. There's a 50% chance, right? Because each square is 25, and a 25% chance of little a, little a. So the most likely genotype, the most likely outcome is big A, little a. And since they're both different, we use the word heterozygous. So the most likely outcome would be heterozygous. If the allele, number one, it's related to sex. Males have it more often, so it'd be sex-linked. And remember, the number one and number two might not be in the same order. Are yours in different order, or is yours in the same order as mine? Different? Some same, some different? Okay. Let's look at this question right here. If the allele, big A, codes for large, right? So if it has this, the flower is going to be really big. If it's lowercase a, the flower is going to be really small, okay? What is the probability that the offspring will have small flowers? Okay, so since the capital A is dominant, this is going to be a big flower because it's got two dominant genes. This flower is also going to be big because this flower has at least one dominant gene. This flower is also going to be big because it also only has, or also has one dominant gene. So this one right here only has homozygous recessive. Homo meaning same, recessive means it's the more rare. And so this flower would be small. So that's 1 out of 4 or 25%. So 25% chance that this cross would produce a small flower. We go down here. Next one, got the same setup. It's just asking something different. Now it says, what is the probability the offspring will be heterozygous? Remember, heterozygous, hetero means different, right? So that would be big A, little a. So this is homozygous dominant, homozygous recessive, and I see 1, 2 heterozygous individuals. So that's 2 out of 4, or 25 plus 25, which gives us a 50% chance. 50% chance they'd be heterozygous. Here we got some peas. And clearly, the big G is dominant for green because even though these peas are heterozygous, that green gene overpowered the yellow gene, and the green peas, well, they're green peas. What is this an example of? Well, this is like the first type of genetics we learned about. It's simple or complete dominance. Sometimes it's known as Mendelian inheritance because Gregor Mendel was the scientist who discovered this, right? He actually studied pea plants in a monastery. So this would be complete or Mendelian dominance, or simple. Sometimes it's called simple. Because it is. It's pretty easy to understand. There's one dominant gene and one recessive gene. Now this looks a little scary, but this is just talking about blood type, right? And I kind of like this chart, right? It shows you, how do you have type A blood? Well, you could be either homozygous, right? Having 2 genes for type A blood, or you could be heterozygous. And since the gene for A overpowers O, this person's got type A. So only one way to have type O blood. You have to have both the homozygous recessive. And it says, what is this an example of? Well, anytime we talk about blood type, Look at how many alleles there are. IA, IB, lowercase I. There's so many alleles. So this is actually called multiple alleles. So this is an example of multiple alleles. That's blood type. Anytime we talk about blood and genetics, we're talking about multiple alleles. Okay, so now we got to work out one of these problems, right? So we're going to make a Punnett square. Color blindness is sex linked. And anytime I see sex linked, I know, okay, it has something to do with gender, male and female. So it has something to do with their biological sex. So we got our female and our male. What are the potential phenotypes? And so that would be like, what do they look like? So we're pheno, physical, what are they going to look like for a cross between a male with color blindness and a carrier female? So we need to make some alleles. So let's do color blindness. Let's do B. So big B, they can see color, and little b is they are colorblind. So they can't see red or green. So the male is colorblind, and since colorblindness is only carried on the sex chromosomes, I'd put a little b on the male's X chromosomes. This person can't see color. The female is a carrier. So what does that mean? In genetics, that means that the person is normal. So this female can see color, no problem, but they are carrying the gene, right? So she can see color, but she's carrying the gene. So what if they have a daughter? Well, this daughter right here would see color and would be a carrier. So we got one carrier. This son could see color just fine because he got the normal gene from his mother. This daughter down here would be colorblind because she got the colorblind gene from her father and from her mother. So we have one colorblind female. And then this male would be colorblind as well. So what we end up with is we got one carrier female, one colorblind female, one normal male, and one colorblind male. So that looks like a pretty good answer to me right there. Snapdragons are actually a type of flower. They're really pretty that have three colors, red, pink, and white. There's actually a lot more, but we'll just focus on those simple ones. And they display a pattern known as incomplete dominance. Anytime you see this word incomplete, that's when you get blended intermediates. So the colors and the shapes and stuff will kind of blend together. So if a gardener were to cross-pollinate two pink flowers, what would the potential outcome be? Well, in these cases, with incomplete dominance, usually we use alleles or symbolic representations like this. Like R might code for red, and then we do like R prime or something like that would be white. And so if a flower has both R and R prime, they'd be pink. So the question's asking, what will happen if we cross-pollinate two pink flowers? So here's one of my pink flowers. And the reason the flower's pink is because of incomplete dominance. I guess I should have put those. Let me erase that real quick. Be a little bit more consistent. And we cross another pink flower. And that flower would be red. This flower would be pink. Another pink. And then this flower, R prime would be white. So we got 25% red, 50% pink, right? These guys would be pink, and 25% white. So 25 red, 50 pink, 25 white. So here we got another one talking about pea plant heights. So we see a capital, lowercase. So I know this is easy, simple, complete dominance. So big T is tall. Little t is small. And it says, if a scientist were to cross two short peas, what would the potential genotypes be? And if I ask about genotype, I'm asking, what kind of genes are they going to have? Homozygous dominant, heterozygous, homozygous recessive. So the only way for pea plant to be short is for it to be homozygous recessive. And do I even really need to go any further? Well, no, because the only genes these parents have to give are short genes. So you're going to have 100% short plants. And since all of them are little t, that's homozygous recessive, right? Homo means the same, right? So they're both the same and they're recessive. So 100% homozygous recessive. Okay, now we got a little family tree, pedigree. And now we have to decide, okay. Is this autosomal or is it sex-linked? And then, is it recessive or is it dominant? If it affects men, the squares, and women, the circles, equally, we call that autosomal. If it affects men more than women, it's sex-linked. If it skips generations, if it skips around, it's recessive. If it shows up in every generation, then it's dominant, right? So let's look. Do I see anybody with the disease in generation 1? No. So I automatically know that this is going to be some kind of recessive trait. So now I'm trying to decide, is it sex-linked or is it autosomal? Well, are men and women affected equally? I see 1 man, 2, 3, 4. I see 4 men with the disease, and I see no women. So I see more men than women. So this is going to be sex-linked recessive. Skips generations and affects men more than women. Here we got another one, another pedigree. The solid means they have the disease. The little half shade means they're carriers. And so again, this skips a generation. This person's got the disease, and this person has the disease. So it skipped the generation. So I know it's recessive. So I know it's going to be one of these two, but I don't know which one. Now I need to compare the sex. Does it affect men and women equally, or is it affecting men? Now, in this situation, no men have the disease, only women. So that still means it's probably 100% of the time going to be autosomal. Because remember, if it's sex-linked, you're going to see a ton of men with it. So this is autosomal recessive. And this is sickle cell, the disease we'll talk about tomorrow. Which of the following statements is true? An individual's collection of genes or alleles is known as a genotype. An individual's phenotype determines genotype. The observable characteristics of an individual are known as genotype, or an individual's collection of genes is known as a phenotype. That is not correct, right? Your collection of genes is known as a genotype. The observable characteristics known as genotype, no. What you can see is phenotype. An individual's phenotype determines genotype. No, the color of your hair is not what controls the genes. The genes control the hair. So the best answer is individuals' collections of genes or alleles. There's a little word we use to represent the symbols, the A's, the B's, the W's, is known as the genotype. Which of the following statements is true? Human offspring need more than one identical copy of a gene. Human offspring need half the number of genes as the parental generation. Human offspring inherit one allele from each parent for each gene. That sounds like a good answer. The other ones sound kind of bogus. 50% of your DNA comes from dad. The other 50% comes from mom. So you actually get one gene for hair color from dad, one from mom. One gene for eye color from mom, one from dad. Just like that for each of your genes. So that's the best answer. Match the genotype, all right, with the correct term. If it's big T, that is homozygous dominant. Homo meaning the same, dominant referring to the fact they're both capital. What about this one? They're both different. And in science, hetero means different. So this would be heterozygous. This next one, if the allele B codes for black and lowercase b codes for white, what phenotype would be displayed? So what would it look like if it has this condition? Well, this is heterozygous. And in this example, since we're dealing with big letters and little letters, the big letter overpowers the lowercase letter, so you can't see it. So this little bunny rabbit would be black. What pattern of inheritance is generally associated with phenotypes being determined by many genes? Examples, human skin color and height. So here's the giveaway right here. Many genes. And in science, another word for many is poly. And you could say gene or genic. So that's why if you were to measure every human on planet Earth, you'd get a graph that looked like this. Most of us are average. There are some extremely tall people and an equal number of extremely small people. That's polygenic, many genes. Okay, we're getting there, y'all. Color blindness is a sex-linked recessive trait. So anytime I see sex-linked, I know that we are dealing with sex. So we got to have the female labeled and the male labeled. And again, let's go back to the big B. They can see color. Lowercase b, they're blind. So we have a colorblind male. So the male is carrying that gene, and the female's normal. She is homozygous for that dominant gene to see color. So what's the outcome? Well, this daughter is a carrier. This son can see color just fine. This daughter is a carrier, so she can see color just fine, but she is carrying the gene for colorblindness. And their other son, right, have no problem seeing color. So we have 2 carrier females and 2 normal males. Uh-oh. Did I make the answer key wrong? Well, my mistake. So the answer, which I don't see here, that was probably my mistake when I was typing it up, should be 2 carrier females and 2 normal males. And I don't see that. So I'm going to go ahead and scratch this question. I'll make sure to give everyone a point back. Let me make a note of that. My apologies. Just randomly pick something. So for that one, since none of the answers match up, just pick one and I'll give them points back for it. Okay, next one. The snapdragon flower is red, pink, white. So we already talked about this. When you deal with these kind of weird blending colors, it's when you got like the R and the R prime. So that'd be red. That'd be white. And so if you have one of each, you'd be pink. So what do we get when we cross a red and pink flower? So the red flower is RR and the pink flower is R prime. So this flower would be red. That flower is red, so 50% red. And then 50% pink. So 50% red, 50% pink. We're good to go. Okay, species of bird feathers can be black, white, or black and white. So anytime we have got both colors showing up at the same time, this is known as codominance. And so obviously B would be for black, W would be for white. And so the bird has both. It's B and W. They don't blend together to make gray. Codominance. So what if we breed a black and white bird with a black and white bird? So one bird there, it's got the genes for both black and white feathers, and then this bird is also black and white. So that bird would be black, so 25% black. This bird would be black and white like the parents, as well as this one. And then our fourth and final potential offspring would be white. So 25% black, 50% black and white, and 25% white. Which technique is used by scientists to produce DNA fingerprints? Well, we actually put DNA, chopped up DNA into a gel, and then we zap it with electricity. and that's known as gel electrophoresis. Gel electrophoresis, and it produces, right, that banding pattern, right? And so if you dumped the DNA here, this would be the smallest fragment. This would be the largest, or sorry, this would be the largest fragment because it didn't travel very far, and this little small fragment traveled really far. And then you could match it. You know, if you had DNA from a crime scene, it should match perfectly, right? And that would be like, oh, this person's a match. This person's the murderer or the criminal. Which of the following statements accurately describes a benefit of a genetically modified crop? And one of the most important things that scientists have done is they've genetically modified crops to be more resistant to pests and disease. So the best answer is this one: increasing disease and pest resistance, so that the corn doesn't get gobbled by beetles, or so that the potato plant doesn't become infected with water mold. We can genetically engineer them to be more resistant. Genetically engineered bacteria can produce human insulin for the treatment of diabetes. How is this accomplished? Well, we take the gene for human insulin and we combine it with the bacteria, the bacterial plasmid, that's what we call it, circular DNA, and we put it back into the bacteria. And then the bacteria starts to make human insulin, and we can take it from the bacteria and give it to the patient. So that first one is the best description. Okay, here we have this DNA fingerprint. We got blood stain. We got Bob, Sue, John, and Lisa. Whose blood is a match? So to do these, all you need to do is match the bars. So the first bar obviously matches all of them. The second bar looks like it matches Bob, Lisa, and John. The second bar doesn't match Bob, so Bob's innocent. Sue was innocent because that bar didn't match. This bar matches John and not Lisa. So if we have to make sure that one matches John, matches John, matches John, matches John. So John is a perfect match. So John is going to jail. Okay, identify the father of the baby using the DNA fingerprints below. So for the baby, right, the DNA is either going to come from mom or dad. So this DNA came from mom, so we don't need to look at that one. These two came from mom, so we don't need to worry about those. And this piece of DNA came from mom. But where did the rest of the baby's DNA come from? Had to have come from dad. So where did this come from? Looks like it came from male number 2. This piece had to come from somewhere. It matches male number 2. This piece matches male number 2. And this piece matches both of these gentlemen. But again, it's got to be a perfect match. So I see 1, 2, 3, 4. And so male number 2 is the child's dad. Okay, so which application of biotechnology is best represented by this diagram? You've got a new gene or putting it in a virus and injecting it into a person, and that virus is injecting that new gene into the nucleus. This is known as gene therapy, and we can use this to replace bad genes with good ones. So the best answer is right there, to use gene therapy to replace faulty, so that means not working genes, with functional ones. What is the significance of the cell type collected by scientists using somatic cell nuclear transfer for therapeutic cloning? When they cloned Dolly, they had to get a somatic cell. They have to get a body cell because the body cell has that diploid set of chromosomes. It has a complete set of DNA. So the best answer is right there. The body cells have a diploid set of chromosomes, which means that if you're trying to clone Dolly the sheep, you're going to have a complete set of information. If you tried to clone something with a gamete, an egg or a sperm, you only have half of the DNA. It's not possible. Which of the following statements is true? Bioremediation uses plants, fungi, and microbes to improve environmental conditions. That right there hits the nail on the head. Bio means life, like plants, fungi, and microbes. Remediation means to clean, to fix, to repair. What essential biomolecules are needed to prepare DNA for gel electrophoresis? That's restriction enzymes. We need those to chop the DNA up into small pieces so that we can get them to move through the gel. So here we've got another gel electrophoresis, right? We've got some deer species here. And which of the deer species below would be considered the closest living relative? So if you're related, you're going to have lots in common. So let's see. I see 1, 2, 3, 4. Okay, and then we got 1, 2, 3, right? So right now this person's got 1, this person's got 2, 2, got 1 and 1. So it's another strike for 3. Another one there, and one there. We got a match for number 2. Then we got another match for number 3. So species 3 is the closest relative to species 1 because they have more DNA in common. They're all deer, right? So they're all going to be related, but this one is the most related because it shares the most amount of DNA. The more DNA and protein you have in common with something, the more related you are. Scientists who work with restriction enzymes and DNA need to closely monitor pH when conducting gel electrophoresis. What can they use to protect these molecules from being denatured? What do our bodies make in nature to protect from changes in pH? Those are buffers. Buffers. We learned about it way back when in the biomolecules unit. Buffers protect proteins and DNA from acids. Why are bacteria perfect candidates for genetic modification? And the best answer is because they reproduce via binary fission. We talked about that during cell division. They make clone copies of themselves. So if you genetically engineer one bacteria and it survives, it'll clone itself and it'll clone that modified DNA, that recombinant DNA. Why do we all need to study science? So we can make informed, fact-based decisions. That is the answer right there. All right. That's all I have for the Zoom. Once you've completed and submitted your exam, it would be in your best interest to go to Unit 8, which I'll actually drag to the bottom, and start working on the Unit 8, 8.1 vocab.